All right, everybody, welcome to the ELISA Lab testing day. The video today is going to be a little different because this lab involves a lot of periods where you have to sit and wait for 5 minutes while things are binding onto other molecules. So what you're going to see here, the slides will be obviously on the computer screen in front of you, and we will be talking through little bits of information as we wait. The ELISA Lab itself is going to be performed right here. This is what's called a well strip. It has 12 little tiny spaces in there that are labeled with the different samples that we're going to be testing. Right now, there are 3 of them that are labeled with a plus. Those will be our positive control. The next 3 here are labeled with a minus. Those will be our negative control. And then we have 2 samples, patient samples, that we'll be analyzing. One of them came from person 3, and the other one came from person 12. So that's these last 3 wells. Before we start here, I just want to review a little bit of vocabulary. What we're testing for here, remember, are antibodies. And we talked about this yesterday. Antibodies are proteins made by your immune system. In this image, they're represented as these purple Y-shaped things. And just like other proteins, their structure is really important to determining their function. Those proteins, you can see here, each antibody is specifically shaped to bond with what's called an epitope. That's one little molecular structure that's sticking off of one side of an antigen. And the antigen is the trigger that's causing the immune system to react. So it could be a spike protein on a COVID-19 virus. It could be a particular carbohydrate on the outside of a bacterial cell. It's some molecular structure that's causing your immune system to respond. So antibodies are particularly designed to bond to that epitope, to that particularly shaped little molecular clump. In this lab, we're testing to see whether these 2 patients, person 3 and person 12, have antibodies to COVID-19. Just so you remember, this is all simulated. The procedure is exactly the same, but there are no human antibodies in this lab. There's definitely no COVID antigens here, and there are not real COVID antibodies. So this is all simulated, but the procedure is exactly the same as what they use to test for COVID antibodies. So the way that this lab works is we put a series of different substances into these wells, and we give them a little bit of time at each step to bond to the previous substance that was added. Right now, I have already put a little bit of the antigen. So this would represent a spike protein from COVID-19, if that's what we were testing for. We put that in every single well here so that it can bond to the bottom of each little well. This has been sitting in there for about 5 minutes. So now it's time to clear this one out and move on to the next step. Every time that we have to add a new substance, the first thing that you'll see me do is I'll take this off and I'm going to kind of dump it on a paper towel here and then just tap all the liquid out. And as I tap it, I kind of move it back and forth so that we don't get any cross-contamination. Once we tap out that liquid, then we need to wash it. The point of this step, which we will do many times through this lab, is to get rid of any extra substances, any molecules that did not bind to these little plastic wells. We want to wash those out. Notice that I'm being really careful not to touch my pipette to the wells themselves. So I'm going to fill all of these to wash these out. This is a lab where it's very important to keep everything sterile and make sure you don't cross-contaminate, or it's very easy to get inaccurate results. So we wash that all out. And then we are going to dump that onto the paper towel and tap it out again. This is a clean side of my paper towel. And you will see me swapping these paper towels out a bunch as we go through and do this today. Again, to prevent cross-contamination as much as possible. So we tap that out, and then we're going to wash it one more time to make sure we absolutely got everything out of there. So give me a second to fill these out. We also want to be sure not to overfill them. If they overflowed into the next well, that could also be a source of cross-contamination. One more dropper here. There. And then once again, we're going to tap this out over here on the side. Every time I do this, I am using a new part of my paper towel and some fresh additional paper towels to make sure that we are not cross-contaminating things over here. All right. Now the next step, as you can see on the screen, is to add the primary antibodies. So now at this point, there should be COVID-19 proteins. And remember, that's not really what they are. That's just what I'm going to call them because that's the simulation that we're doing today. There should be COVID-19 proteins bonded to the bottom of every single well in this strip. So now what we want to do is we want to add our samples in, because the point of this is to test and see whether the samples have the antibodies that we need. So our first sample that we're going to add is actually our positive control. For these, I'm going to be using a micropipette, that's this little tool right here, with a clean, sterile tip on it for each sample. So this vial contains a positive control. It's labeled with a plus. So that means I know that this vial does contain the antibodies that we want to test for. So I'm going to put a little bit of that into each of these wells. 1, 2, 3. Oh, that was too much. Hang on. 3. Then I am going to change the tip on my pipette and get a clean one. Next, I'm going to put in the negative controls. The negative control is a sample that we know does not include the antibodies to this particular protein. Positive and negative controls play really important roles in tests like this because they help you to determine whether your results are reliable. Because you want to make sure the negative should give you a negative result, the positive should give you a positive result, and if either of those does not happen, then something was not working correctly. I'm going to grab a new clean tip, and now I'm going to add my patient samples. So the positive and negative control, we know the plus should have antibodies, we know the minus should not, but these two patient samples are our mystery tests. These are the ones where we don't know whether these people have been exposed to this disease and whether their bodies have developed the antibodies to it. And so that is what we are testing for today. We are testing each of them 3 times for the same reason that you do any other scientific experiment multiple times, so that in case one of them gets contaminated, we still hopefully have some reliable data to look at. One final clean pipette for the sample from person 12. And then again, we are going to give this 5 minutes for these substances to bond if there is antibody present. And there's the last one. And I'm going to start a timer for 5 minutes. So we added our positive and negative controls, we added our 2 patient samples, and now we're going to incubate it for 5 minutes. What we're testing for here is to see whether each of these samples contains the antibody that has that particular specific shape to bond with the antigen that we already put in there. So in this case, we're testing to see whether each of these samples has the antibodies that will bond to that COVID-19 spike protein that triggers the immune response. If those antibodies are present in the samples from these 2 people, right now they should be floating around in there and bonding onto those proteins that are already bonded to the bottom. If there are no antibodies in there, then obviously nothing is happening. And so we're just going to sit for a few minutes and wait to see whether those antibodies are there. All right, it has now been about 5 minutes that those samples have been in there. So there's been some time for the antibodies, if they are present, to bond to the antigen on the bottom. So now we're going to empty this out and then wash it twice again. I'm going to wash this out with my wash buffer solution once again, making really sure not to touch the pipette to the thing. We're going to fill those just about to the top. after we wash this, the next substance that we're going to add is called a secondary antibody. This was an antibody that was developed in a laboratory so that it will bond to the primary antibody if it's there. And this secondary antibody, hang on a second. This secondary antibody has an enzyme that is attached to it. And that enzyme is going to help us detect and visibly be able to see whether these molecules were present by the end of this test. So I'm washing it out one more time. There's some bubbles forming in there. All right. So that was my second wash. So now I'm going to tap those out, dump that wash material out, get all of that liquid out of there. And now this time, oops, I have once again a clean pipette tip, and now I'm going to add the secondary antibody into all of the wells. Hang on, just making sure I'm adding the right amount here. Even with this pipette tip, I'm trying to be careful not to touch it to the sides of the beaker. Or sorry, of the wells on this well strip. As I put this into every single tube along the way. And this will also have to sit for 5 minutes. And I will show you a little bit of what is going on in here. One more. All right. I'm going to discard that pipette tip. So we washed it out twice with a buffer. And then we added, oh, I got a little bit ahead of myself. Uh-oh. Got to figure out how to go backwards on this. There we go. There we go. So now we're adding our secondary antibody. And what the secondary antibody does, so the secondary antibody was made particularly for this particular test, and it's made specifically for each different ELISA that's developed. And this is a secondary antibody that has a particular shape that's made to bond to that primary antibody, if it's there. So remember, if that first sample had antibodies, they bonded to the antigen. And now if those primary antibodies are there, the secondary antibodies right now, while we're waiting our 5 minutes, are grabbing onto those. Hang on, restarting my timer. There you go. So our secondary antibodies are grabbing onto the primary antibodies if they're there. If not, these secondary antibodies right now are just kind of floating around there, and we will wash them all out when we do our next washing step. The secondary antibody is linked to an enzyme called horseradish peroxidase. That's the enzyme that's used in this particular test. But it's an enzyme that's actually attached to each of these secondary antibodies. And that's the E part of ELISA, enzyme linked. And that enzyme is what's going to allow us to see a color change in the next step, which will be the final step, to actually gather our data and determine whether or not those antibodies were present. So we're going to wait a few more minutes here, and then we will come back, wash it out once more, and then add the substrate for that enzyme to act on so that we can actually read the data that we get as a result of this. So our secondary antibody has now had about 5 minutes to bind to the primary antibody if there were primary antibodies in the sample. So we're going to dump this out, and this time we're going to wash it out 3 times. I'm going to make really sure everything's out of the way for this final step. Okay, so I'm going to dump all of that, and then I'm going to get my wash buffer. Whoa, try not to squirt it all over my computer. All right. At this point, it's really important to wash out any extra antibodies from any of the steps, any of those extra molecules that are floating around, because the next step here is going to be to add the substrate of the enzyme. And we want the only enzyme in there to be the enzyme that has linked on to the primary antibody, that has linked on to those proteins. And so we want to make sure that we wash everything else out really well. So this is going to take me just a minute to wash this out 3 times. All right, that's the first one. I'm going to dump and tap it out. Then wash it once more. This is the second time. Sometimes this step gets a little bubbly because there is an actual like detergent type substance. We're washing things, so it is basically like soap, to make sure that all of those extra things get washed out of there. So that's why sometimes it gets a little bubbly. All right, just about done with the second wash here. All right, clean paper towel for my tapping out. And one final wash with this buffer solution. All right, clean paper towel. And before we add this final substrate, I'm just going to swap these out so that my computer stays dry and so you can see a little better. So the final step here is going to be to add the substrate of the enzyme. So I have one more clean pipette tip, and I have my enzyme substrate right here that I'm going to add into all of these little wells. And this could take a few minutes as the reaction happens. We need to give it a couple minutes to change color, but some of them could happen pretty quickly too. Couple more. There. So now what's happening, and you might be able to see that our positive controls are already starting to change, so we'll be able to read this in just a minute. we're adding the substrate that the enzyme we put in last time acts on. And we're waiting to watch for a color change. So remember, the antigen was what we put in there first. That would be our COVID spike protein. Then we added our samples to test whether that primary antibody is there. That's really the question. Is this piece there or not? If not, then nothing else happened because there was no primary antibody for any of the rest of this to bond onto. But if that primary antibody was there, our secondary antibody bonded to that. And remember, the secondary antibody had this enzyme attached to it. And now we just added the substrate for the enzyme. And so if all of that's there and if that enzyme is present, then that enzyme will catalyze a reaction that will turn that substrate blue. That's the color change that we're looking for. So it looks like even though it hasn't been 5 minutes, we can already start gathering our data. So I'll try and hold this a little more close to the camera so you can see. So you can see our 3 wells with the pluses on them turned blue, which they should because a positive control has the primary antibody in it. That's what it is. The negatives stayed clear, which is also what should have happened because we know that that was not there. Then we had our 2 patient samples. These wells right above my finger are patient 3. So far, they have stayed completely clear. There's no color there. But these 3 wells are from patient 12, and they are turning blue, which means the antibody was present in patient 12, but not in patient 3. Just a little more background as you look at this. You're going to answer some post-lab questions here to talk about the results of this and talk a little more about how this works. Think about what we learned yesterday and whether having antibodies definitely means that they were exposed to COVID-19, whether it definitely means they had COVID-19, and whether it means that they have it now. There's a lot we still don't know. Antibody tests are less reliable. And there's also a lot that we don't know. If you have antibodies, scientists still are not sure whether that will completely prevent you from getting COVID again. Scientists also do not know how long those antibodies stay in your body and how long that immunity might last. So there's a lot that we're still learning about the immune response to COVID and our ability to maintain that immunity over time. Those are really important questions as we try and figure out how to treat this and how to come up with a vaccine that might last for an extended period of time so people are protected from this disease for a long time. And this ELISA test is one way that they are testing for these antibodies over time to kind of learn some of those things about how long that immunity might last. In case you're interested, like I said, there were no real human molecules used in here. The antigen that we put down first was actually a protein that came from a chicken. And what they did, the way that they made the primary and secondary antibodies for this lab kit, they took chicken proteins and they injected them into a rabbit because the rabbit doesn't normally have those proteins. So the rabbit's body looked at it and say, hey, what's this chicken protein doing here? And thought it was an intruder like a virus and made antibodies against it. And so then scientists were able to collect those antibodies from the bloodstream of that rabbit. And then they took those antibodies and they injected them into a goat. And the goat's body said, what the heck are these antibodies? They're not supposed to be here. And it started making antibodies against the rabbit antibodies. So that was the source of these for this particular lab. And that's how scientists can develop these ELISA tests to detect particular antigens or antibodies, is using procedures kind of like that to make the molecules that they want for these tests. You're now going to go and work on your post lab, and you don't need to look at that slide too much. That's kind of my notes to myself. The post lab questions will be due in a few days, so you can check in Classroom for the date that those are due, and feel free to contact me if you have any questions. I hope you have a great rest of the day.